Very good evening to all of you. It's a great pleasure for me to announce the opening of this webinar. So it was conceptualized by Dr. Moshumi Saikia, Dr. Sharmishta Chaudhary, Dr. Teresa Bishwas and Dr. Anil Dasgupta. That this time, instead of taking the faculties and consultants as a panelist, we can keep our youngsters who are actually indeed very good speakers, and right from the beginning, if you can give them the proper limelight, they can actually groom much better. So it was our cumulative idea, and the first time we are going to introduce a good number of youngsters, all the postgraduate trainees, the moderators and speakers, all of them. Some of them are very wonderful speakers, some of them with a very dedicated work you can find. And I actually wish all of them all the very best with this inauguration of the panel. And therefore, all the panelists and moderators will be introduced by Dr. Madhurima Bora, who is the chairperson of the Students Affairs Committee of Assam. And I would like to invite Dr. Madhurima Bora to come and give a brief introduction about all of the panelists and the moderator, and let the show begin. We are really waiting very eagerly for this thing to conduct, and all the very best to our youngsters. And I am sure you will rock. And let's have the good evening. Thank you.
Thank you, Dr. Sen, for making me a part of this session. Myself, Dr. Madhurima Bora, Associate Professor, Department of Biochemistry, Guwahati Medical College. I would like to introduce the panelists and the moderator of this session. First panelist is Dr. Soumyamoy Das. He passed MBBS from NSCB Medical College, Jabalpur, Madhya Pradesh in 2005. He is a third-year postgraduate trainee of the Biochemistry Department, Guwahati Medical College. He is an affiliate member of ACCLMP Assam chapter. Our second panelist is Dr. Indranil Chakrabarti. He passed MBBS from Calcutta Medical College, Kolkata in 1993. He is a third-year postgraduate trainee of Biochemistry Department, Medical College, Calcutta. He is an affiliate member, ACCLMP West Bengal chapter. Third panelist is Dr. Somnath Haldar. He passed MBBS from Bardwan Medical College, Bardwan in 2006. He is a third-year postgraduate trainee of Biochemistry Department, Calcutta National Medical College, Kolkata. He is an affiliate member, ACCLMP of West Bengal chapter. Fourth panelist is Dr. Bidyut Bhuyan. He passed MBBS from National Medical College and Teaching Hospital, Birganj in 2011. He is a third-year postgraduate trainee of Biochemistry Department, Assam Medical College and Hospital, Dibrugarh. He is an affiliate member of ACCLMP Assam chapter. Then fifth panelist is Dr. Manidip Chakraborty. He passed MBBS from Regional Institute of Medical Science, Imphal, Manipur in 2003. He is a second-year postgraduate trainee of Biochemistry Department, Silchar Medical College, Silchar. He is an affiliate member of ACCLMP Assam chapter. Then the sixth panelist is Dr. Koushik Kumar Das. He passed MBBS from Calcutta National Medical College and Hospital, Kolkata in 2018. He is a first-year postgraduate trainee of Biochemistry Department of RG Kar Medical College and Hospital, Kolkata. He is an affiliate member of ACCLMP West Bengal chapter. Then our moderator is Dr. Priyanka Moitra. She passed MBBS from Lady Hardinge Medical College, New Delhi in 2003. She is the third-year postgraduate trainee by Chemistry Department, RG Kar Medical Hospital, Kolkata. I welcome all the panelists and I over the mic to Dr. Priyanka Moitra to start the session.
Good evening, everyone. This is Dr. Priyanka Moitro. I would like to take this opportunity to thank ACCLMP for providing us this platform to discuss the role of clinical chemistry laboratory in the diagnosis of common inborn errors of metabolism. What are inborn errors of metabolism? They refer to a group of diseases which affect the ability to utilize nutrients for the generation of energy. They mostly present in infancy and in newborn period, but they might, like those of the fatty acid oxidation, they may present even in adulthood. But irrespective of their age of presentation, their course is very devastating. Hence, early diagnosis and prompt treatment initiation is very important. And on that note, I would like to begin this session by inviting Dr. Koushik Kumar Das to take up the mic and answer the first question. Dr. Das, can you briefly shed some insight on the different categories of the common inborn errors of metabolism that one can encounter in the clinical laboratory? Dr. Das?
Hello. Thank you, Dr. Priyanka Moitro. Am I audible and visible?
Yes. Please carry on.
Okay. First of all, I would like to express my utmost gratitude towards ACCLMP for providing us the opportunity as panelists to speak as the panelist in this panel discussion on the role of clinical lab on the diagnosis of inborn errors of metabolism. Now, the question which is addressed to me, it is towards the brief introduction on the common inborn errors of metabolism. So before going into the detail or stating the names of the inborn errors of metabolism which are commonly encountered, I would like to throw some insight towards what these common inborn errors of metabolism are. So next slide, please. Yeah. So I would like to throw some light on the inborn errors of metabolism from a clinical lab perspective. Now, what happens in a normal person? A substrate is converted into a product through a metabolic pathway. Now, whenever there is hindrance in this metabolic pathway, either due to any enzyme defect or due to defect in transporter, there is hindrance in the metabolic pathway, and that leads to accumulation of the substrate itself and also the deficiency of product. Again, this substrate is bypassed through other metabolic pathways and other metabolic byproducts are produced. So in a nutshell, in inborn errors of metabolism, in clinical lab, what we see is the accumulation or increased level of substrate, decreased level of the product, and increased level of the byproducts. Now we are going into the detail of the classification of the inborn errors of metabolism. Now classifying them, we can classify them under 4 major categories. These can be disorder of metabolism, carbohydrate metabolism, and disorders of lysosomal storage. Now, in the amino acid metabolism category, we can have amino acidopathy. Well, these diseases occur to defect metabolism of amino acids, and that leads to accumulation of the specific amino acids. Now, as a result of that, there is also accumulation of certain organic acids as well, which are the intermediates of amino acid metabolism, and that leads to the second, which is organic acidemias. And the amino acid metabolism defects, they can be due to certain enzyme defects or defects in the enzymes of urea cycle that leads to urea cycle disorders. Now the fatty acid oxidation defects, well, they occur due to the deficiency mainly of medium chain fatty acyl-CoA dehydrogenase or long chain 3-hydroxy fatty acyl-CoA dehydrogenase. Fatty acid oxidation disorders can be again due to defective enzymes of peroxisome, and that are peroxisomal disorders. Under carbohydrate metabolism category, most commonly we encounter classic galactosemia. We also see glucose 6-phosphate dehydrogenase deficiency, and we see all the glycogen storage diseases as well. Now, can we move on to the next slide? Now we are going to enumerate all the common inborn errors of metabolism that are encountered in clinical lab. Next slide, please. So under the amino acidopathy, what we commonly get is the phenylketonuria. Now this occurs due to deficiency of phenylalanine hydroxylase, which leads to accumulation of the amino acid phenylalanine. We get tyrosinemia. There are 3 classes of tyrosinemia. Among them, the type 1 is the most fatal, and all these tyrosinemia, they occur due to defects in the metabolism of tyrosine amino acids. We also get homocystinuria due to defective metabolism of the amino acid methionine, which leads to accumulation of homocysteine. We also commonly see maple syrup urine disease that occurs due to defective metabolism of branch chain amino acids like leucine, isoleucine, and valine. Next up, under the urea cycle disorders, we commonly get citrullinemia and argininosuccinic aciduria. Now, these disorders are caused due to argininosuccinate synthetase and argininosuccinate lyase enzyme deficiencies. Under organic acidemias, the most common one is glutaric acidemia that occurs due to defective metabolism of the amino acid lysine, hydroxylysine, and tryptophan. We also see propionic acidemia, methylmalonic acidemia, and isovaleric acidemia. Under the peroxisomal disorders, which I mentioned before, the peroxisomal enzymes, they are involved in the degradation of certain complex biomolecules. So now these peroxisomal enzymes, they are involved in the oxidation of fatty acids. So the most common one, which we encounter, is the X-linked adrenoleukodystrophy. Now the next slide, please. Now under the carbohydrate metabolism defects, we commonly encounter glycogen storage disease, among which type 1 and type 2 are the most common, like von Giex and Pompe's disease. And others I have already mentioned, classic galactosemia and glucose 6-phosphate dehydrogenase deficiency. Fatty acid oxidation defects I have already mentioned, and lysosomal storage diseases, where lysosomal enzymes, they're responsible for the degradation of complex biomolecules, and defects in those enzymes leads to accumulation of those specific complex biomolecules. The most common ones are Gaucher's disease or mucopolysaccharidosis type 1. So in a nutshell, these are the common IEMs or inborn errors of metabolism that we encounter in clinical lab. Thank you.
Thank you, Dr. Koushik. That was a very good insight on the classification of the IEMs, the common IEMs that we encounter. Thank you very much. My next question is to Dr. Indranil Chakrabarti. What do you feel, sir? Do you feel that clinical laboratories need to develop a robust screening policy for prenatal diagnosis of the common inborn errors of metabolism? And if so, then can you please give your opinion on what should be the constituents of such a policy? Dr. Indranil. Dr. Chakrabarti? Yes. Dr. Chakrabarti?
Am I audible?
Yes, you are audible, but your video is not visible. Please switch on your video.
Here it is, the host has disabled the video. Mr. Anil.
Mr.
Please.
Anil, kindly enable the audio for Dr. Indranil. Try clicking on it again. Anyway, you can start. He will enable the audio. You please start answering the question.
Thank you, Dr. Priyanka.
Yes.
Thank you, the organizers of ACCLMP Con 2020, for giving us opportunity on a national to talk on a national platform. Now, thank you once again, the organizers of ACCLMP Con, for such wonderful topic of panel discussion, because we are not far away from 21st of November, because 21st of November is marked as IEM Day in India to raise awareness about the inborn errors of metabolism. Now the question, what Dr. Priyanka has asked me, why do we need such robust screening policy for IEM? Now, before saying yes I must share some important informations, obviously in relation to Indian scenario, that 1 in every 500 to 1,000 newborns was observed to suffer from inborn errors of metabolism. India accounted for a staggering 29% of the global deaths of newborns on their very first day of life. Next slide. Next slide, please. 5 to 15% of newborns are sick due to metabolic diseases in India. And Dr. Koushik Das has already summarized different diseases in nice tabular form. And today, India accounts for the greatest burden of newborn deaths in the world. Next slide, please. And therefore, considering these informations and these data, I must say, yes, clinically, there must be a robust screening policy for screening of IMs in the clinical labs. Now, coming to the second part of my question, what should be the components of such screening policy? First of all, I must say that the screening policy must be initiated from the government level, central as well as state government. And we all know that some of the states in India has already installed NBS or newborn screening policy mandatory in the states like Goa, Kerala, Chandigarh, etc. And following the discretion of the, following a part of the, following the discretion of national policy or a part of the national policy, we must fix the target population where the screening to be done. The target population definitely for prenatal diagnosis, the pregnant mothers. Then time and frequency of screening, when to screen and how many times to screen. I think every pregnant mother should be screened from the very beginning of gestation period. And regarding the frequency, I think at least 2 to 3 times in the entire gestation period. Now, coming to another part of a policy, that is, it should be mandatory screening. It should be mandated to screen of any unexplained metabolic acidosis, hypoglycemia, ketonuria, etc. pregnant mothers, which are very common under and definitely under high risk profiles. Next slide. Now, coming to the human resources in the IM lab, because IM lab must have skilled technicians to carry out sample preparation, monitoring of the lab machines, and they have to deal with some micro techniques like microfluid based enzyme assays, etc. So lab technicians must be highly skilled. Now, coming to installations and instruments, every basic IM lab, I think, must have spectrophotometer and GCMS instrument, because they play a big role in the diagnosis of common IMs in the clinical lab. Now, finally, in the final part of our policy, everybody should follow the state models where IM screening or newborn screening has already implemented. I have already stated in the beginning of my talk, like Goa, Kerala, Chandigarh, etc. They have already implemented, so they are highly experienced. And if we have to start in every state, start the same policy in every state, in the every clinical lab of the government hospitals, and sometimes there should be some collaboration of government hospital with the private hospitals regarding the universal screening of IM. Thank you. Thank you.
Thank you, Dr. Indranil. That was a very staggering statistics that you presented regarding the children born with IM and the policies that you have outlined. They are very robust. Now, let us move on to our next speaker for the day. That is Dr. Soumyamoy Das. Dr. Das, my question to you is, can you enlighten us on the role afforded by simple biochemical tests while screening for common inborn errors of metabolism? Dr. Das, over to you.
Thank you, Dr. Priyanka, and thank you, the organizer of ACCLMP Con, for providing us this opportunity today. So, first of all, I want to tell that there are many diagnostic tests for the diagnosis of IEM or inborn error of metabolism. These are available in high-end laboratories, but in a routine laboratory, there are few tests which can be done for the screening of most of the common IMS. So we know that most of the metabolic products which are excreted from the body in the urine. And so first of all, I want to mention a few tests which are routinely done in a clinical laboratory. First of all, the ferric chloride test. That can be usually done in a laboratory, and for this test, one should not go to a sophisticated class. So in ferric chloride test, one can screen for the phenylketonuria or maple syrup urine disease or histidinemia or alkaptonuria. If the test, I'm just want to say a few tests, how to do this test. Like ferric chloride test, in 5 ml of urine that is collected from the newborn, a few drops of ferric chloride is added. If phenyl pyruvate is present in the urine, then the test will come positive. There will be a color change. In case of phenylketonuria, that is a greenish blue color. So in first case, we can screen whether the baby is suffering from phenylketonuria or not. After the screening test, one can subject this sample for further testing facilities. In other testing like ferric chloride, by ferric chloride, we can screen the alkaptonuria and all these things. The second test is the Elsian blue test, Elsian blue spot test. It is a screening test for mucopolysaccharidosis. Here, in mucopolysaccharidosis, when urine of the baby is added in a 10 ml of urine, few drops of Elsian blue reagent is added. If the test comes positive, that will turn blue on a white background. That is very easily available in a routine biological laboratories. Other tests like Benedict test for the measurement or measurement of reducing substrates like sugars in galactosemia, in fructosemia. Normally they are present in the urine, but they are present in a very little amount that cannot be measured. And so if the patient is suffering from these diseases, that can be easily measured by Benedict test. Other tests are like cyanide nitroprocesside test for homocystinuria, or the in case the child, if child is suffering from glycogen storage disorders or organic acidemia or tyrosinemia, then the lab physician can perform the dipstick for the identification of the ketone bodies in the urine. Next slide, please. Thank you. In this slide, I want to show the one test that is called Guthrie's bacterial inhibition test. That test was first devised by Dr. Robert Guthrie after the birth of his own child who is suffering from phenylketonuria. In this test, capillary blood is collected from the heel of a newborn, and that were collected in a card, and on the initial days, the punch out of the dried card was inoculated in a petri dish containing the Bacillus subtilis and a inhibitory substance like the beta 2 phenylalanine. The beta 2 phenylalanine is an antagonist of phenylalanine. If the blood contain phenylalanine, then there will be a growth of the bacteria, as bacteria is phenylalanine for its growth. But if the blood contain lesser amount of phenylalanine, or the baby is not suffering from phenylketonuria, then that antagonistic the phenylalanine, and there will be no growth. That is the first test in the diagnosis of phenylketonuria. So we have seen, we can see in so many screening tests can be performed in routine laboratory settings before proceeding to the advanced testing facilities. So these screening tests are usually available in a routine biochemical laboratory. So before going or before subjecting these urine or blood to for higher tests like genes in next generation sequencing or Sanger technique or mass spectrometry, one can, for the screening purpose, can do these simple tests that are easily available, and these are also low cost tests. Thank you very much.
Thank you, Dr. Das, for outlining those very fast, quick, and simple, cost-effective biochemical tests, which can be done in every laboratory. Thank you so much for your insight on this matter. Now let us move on to the next question. And for this question, I would like to call upon Dr. Bidyut Bhuyan. Dr. Bhuyan, can you provide us some insight on the role of genetic and molecular tests in the prenatal screening protocol of inborn errors of metabolism? Because we have just discussed the basic screening tests. So can you please provide us an insight on this?
Yes,
Dr.
yes.
Bhuyan? Thank you.
Thank you for the question, Dr. Priyanka Moitra. And thank you, ACCLMP, for providing us this opportunity to talk on this platform. So I would like to talk upon the role of molecular genetics in the prenatal screening of IEMs. Next slide. First,
Dr. Bhuyan, sorry to interrupt you there. Sorry to interrupt you. Just stick to the salient points. Dr. Bhuyan, just stick to the salient points. Yeah, thank you.
Yes. First, let's see what are the sampling techniques. So first we come across the CVS, or the chorionic villus sampling. This is performed around 10 to 12 weeks of pregnancy. So what do we understand by chorionic villus sampling? Chorionic villus is that when the growing embryo at the early stage, it divides into two things. So one goes to the growing fetus, and another is the finger-like projection which goes into the uterine wall to in order to extract maximum amount of nutrition for the growing embryo. So interesting fact is that this chorionic villus has the same DNA which the growing fetus or the embryo has. So we take advantage of this fact and we can take the sample and perform the test. Similarly, amniotic test is there. We know that it is the fluid that is the baby urinates and the baby swallows. So that is the, if there is any suppose problem in the baby, then the same thing will come out of the baby's urine and we can detect in that amniotic fluid itself. So regarding the fetal sexing, another test, we do it basically for the X-linked diseases which are inherited mainly for the males, mainly by the males. And suppose if the it is done by the ultrasound, suppose if the baby is a female, then we don't further do anything. And if the baby is a male baby, then we go for the further tests. Next slide, please. The amniocentesis is the same procedure by which we poke a needle and we take out the amniotic fluid from by poking the amniotic sac. It is performed around 14 to 18 weeks, and the same same thing which I just talk in the amniotic fluid is done. It's the same things we can do. Then regarding the free fetal DNA, it is the circulating DNA from the fetus that can be detected in the blood of a pregnant woman. It is most importantly, it is non-invasive because we don't unlike before that CVS or amniocentesis, we don't poke anything inside the amniotic sac, so it is harmless. And we just take the mother's blood and we look for the circulating DNA, we do the test accordingly. And if everything, all this fails, then there is another procedure. It is called the chordocentesis. It is a very high risk procedure. Actually, chances of miscarriages are very high. So fetal blood sampling or the chordocentesis is usually prohibited, but if everything fails, then we can go ahead with this test and see for any defects. So quickly, I'll speak about a few important techniques which we do with these samples which we acquire already. Now, like the first important thing is that PCR, polymerase chain reaction. For this, so we need some prerequisites. That is the DNA sample which we already got from the baby or from the CVS or the amniocentesis procedure. Then the primers we can design in the molecular lab. Primers, because we know the human gene sequence, and primers can be designed accordingly. And DNTPs or the free nucleotides are the also required. And Taq polymerase is actually acquired from the Thermus aqueous polymerase. It is a highly heat stable, because normally our human polymerases, they act at 37 degree, but Taq polymerase, it will act at a higher degree centigrade. And there is the mix buffer. So we mix everything and put in the PCR tube and we put in the thermal cycler machine. And by the three main things are there which takes place. Is a denaturing, when the DNA breaks down, strands separate, annealing, that primer will come and bind to the exact counterpart of the this sequence, gene sequence, and then the extension. Then DNA, the Taq polymerase will come, and then there will be extension of the further DNA. So in this way, we can generate a longer sequence of genome to study the diseases better. The next slide, please. Another important procedure is the comparative genomic hybridization. Suppose that we take here in this procedure, mainly we are comparing two genomes, the tumor DNA or that is the defective DNA. Later here I have shown tumor, but it may be any IEM DNA. and the normal DNA. We take from the same baby, then in the metaphase state, we do the hybridization. First we break down the DNAs in the metaphase state, then we stick with the primers, and then we do the hybridization. And then hybridization, this primer will be fluorescent primer. We label it with fluorescent dye, and we then hybridization is done. Then after that, we have a detector in which we can detect this thing. Like there will be two things can be easily caught with this problem, with this procedure. That is the duplication of the genome or the deletion of the genome. So if there is duplication, then the gene, then the primer which we have added will show up more. And if there is deletion, then it will show up less. Then we can catch if there is any problem with this duplication or deletion in the genome. And we can hence catch the disease. Next slide, please. And another important is the spectral karyotyping. Normally, when we see a karyotyping picture, it is black and white. But here we can see it is colorful, because what we are doing basically is we have attached the genome with design primers, which is labeled again with fluorescent dye. So we can color the chromosomes according to our wish. Like here we can see yellow, blue, 1, 2, all the different colors we are putting. So we can, at wish, we can paint the chromosomes according to our wish. And then we can see if there is any defect. Suppose deletion and duplication will not be caught with this procedure. Here, the best thing we can detect is substitution and translocation. By this procedure, we can easily, suppose in the gene 13, here we can see in the slide, the white color is painted with the number 3 and 13. Let's compare. So it should be pink, but actually it is white. So there is some, we can see that in the lower part, there is some translocation from the gene 3. So we can detect in this way the disease. Next slide, please. So now we see, we can finally see the clinical utility of genetic testing. What are the, how it helps the family or the couple. It can help reach a definitive diagnosis when biochemical diagnosis is equivocal or not clear. And it helps in the diagnosis of affected family member carrier identification by targeted testing. And it helps for a guidance of monitoring of a potential future complication. And it also helps avoiding unnecessary diagnostic procedure, tests, and treatments. Thank you so much.
Thank you, Dr. Bhuyan, for that insight into this beautiful images that you have provided on spectral karyotyping and, of course, PCR and comparative genome hybridization, which we know a bit, but spectral is quite new to me. Thanks for sharing it. Those are the tests that can definitely clinch a diagnosis. Now, my next question is to Dr. Indranil Chakrabarti. And the question is that vis-a-vis to screening tests, what in your opinion can constitute a robust confirmatory panel of tests for cementing the diagnosis of common inborn errors of metabolism? Dr. Indranil Chakrabarti.
Priyanka, my video is not the best. Say Anil.
Sir, you carry on. We will request Anil to look into that.
Okay. 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 Am I audible? Am I audible? Am I audible?
You are audible. Yes, you are audible. Please carry on.
Okay.
You are visible as well.
Okay. Okay. Just thank you, Dr. Priyanka. Now, next slide. Okay. Now, in addition to clinical presentation and routine lab tests, and the diagnosis of patients with IM relies on specific tests. Now, we have already discussed, other panelists have already discussed some approaches. Now, I want to highlight how to cement the diagnosis, how to label the final diagnosis of the IM in the clinical labs. Now, this is the LCMS, MS for amino acid analysis. Basically, mass spectrometry is the mainstay to label final diagnosis. LCMS, MS for amino acids, and GCMS for organic acid analysis. Next slide. Now, tandem mass spectrometry, with or without liquid chromatographic separation for acyl carnitine profile, acyl glycine profile, etc. Now, here in this slide, we can clearly view the spectrophotometer, different components, and how does it work. It is very clear from the diagram. There is a ionization chamber, accelerator part, then deflection chamber in the magnetic field, and finally detection on the recorder. And different components are clearly labeled. Everybody can see it and easy to understand. How does a spectrophotometer act? Now, move on to the next slide, please. Now, the combination of these tests using different specimen types is the key to the biochemical confirmation of metabolic disorders. Now, I'm coming to a very new approach in the diagnosis, in the final diagnosis of IMs, that is metabolomics. It is a new approach to the diagnosis of IM, especially when the clinical presentations are non-specific. Nowadays, metabolomics is often termed as new clinical chemistry. That is basically focused on the analysis of set of all small molecule metabolites present in the biological samples. And the slide, previous slide, please. The slide depicts the entire system of metabolomics in a clinical lab, including the operation of the systems. Definitely, it is a complex instrument with data recording systems and etc. Now, it is move on to the next slide. Now, what are the different advantages? Metabolomics, I have already told, that is a new clinical chemistry, and it's a very, very helpful to label the final diagnosis of IM following the basic screening approach. Now, different advantages include measures multiple metabolite at once, and no separation is required. That is amazing. It allows metabolic profiles or metabolic fingerprints. Like DNA fingerprint, we have learned metabolic fingerprint under the head of metabolomics. And analysis and results is obtained in less than 60-second time. That is one of the great advantage of metabolomic study. Next slide, please. Now, MS and nuclear magnetic resonance, that is NMR, are the main analytical techniques. Although IMS, that is ion mobility spectrometry, combined with high resolution MS provides better coverage. Now, metabolomics can also lead to the discovery of new IM, the novel biomarkers, and definitely a better understanding of IMs. And finally, the inherent challenges that are associated with the large amount of data generated by high throughput technologies, including how to track, store, and share the data. It is crucial, next slide. It is crucial to develop advanced bioinformatics solutions comprehensive open access databases, and computer methodologies and software to convert the huge amount of data generated by this approach into effective, clinically actionable tools that can aid in final decision making. That is, for cementing the diagnosis of inborn errors of metabolism. Thank you.
Thank you so much for highlighting that it is a marriage between mass spectrometry, NMR, and metabolomics, and
Right.
advanced bioinformatics for clinching and for confirming, for cementing the diagnosis rather for an IEM. Thank you so much, Dr. Chakrabarti. Let us now move on to our next question for the session. And the question is to Dr. Somnath Haldar. Dr. Haldar, with respect to the common inherited disorders of lipoprotein metabolism, what are the diagnostic strategies that should be implemented by the clinical laboratory? Dr. Haldar?
Thank you, Dr. Priyanka, and thank you, Dr. Organizer of ACCLMP for providing this opportunity to me. And regarding my question, what will be my strategy to diagnose the common inherited lipoprotein metabolism disorder? How will you start my strategy? First, the lipoprotein disorder may be due to primary causes or may be due to secondary causes. As secondary causes are more commoner than primary causes, we should first exclude the secondary causes such as diabetes mellitus, thyroid dysfunction, and nephrotic syndrome, etc. by doing the appropriate test. After exclusion of all secondary causes, we should focus for detection of the primary causes of this common inherited disorder of lipoprotein metabolism. Common laboratory strategy for this should be biochemical assay for detection of different lipoprotein for different lipid parameters, electrophoresis assay for analyzing different lipoprotein bands, and genetic assay for detection of different apolipoprotein gene defects, either by doing polymorphism study or by doing direct DNA sequencing technique, or by doing more advanced techniques such as quantification and detection of lipoprotein receptors. And after doing this, next slide please. For easy understanding of these diseases, and I have divided the diagnostic strategy in 6 common categories. First, I will discuss the diseases associated with high triglyceride level and normal cholesterol level. Familial hypertriglyceridemia. This disease can be diagnosed by a triad of elevated level of plasma triglyceride level and elevated cholesterol level and reduced HDL-C level. It is an autosomal dominant disorder. It is commonly associated with the plasma TG level in between 250 and 100 milligrams, 1000 milligrams per day. Now, the second disorder is lipoprotein lipase deficiency disorder. This disorder may be due to pure lipoprotein lipase deficiency or may be due to deficiency of ApoC, which is a activator of lipoprotein lipase enzyme. For distinguish these diseases, we can perform the post-heparin lipolytic assay. In post-heparin lipolytic assay, we can find the lipoprotein lipase level is low or absent in case of lipoprotein lipase deficiency. whereas in case of ApoC2 deficiency, the lipoprotein lipase enzyme is low but never absent. Fasting PG level is more than 1000 milligram per DL in case of lipoprotein lipase deficiency. And if we add the normal plasma in test heparinase plasma, and then the ApoC2 level will be increased, and this will cause the increased lipoprotein lipase activity. Now the ApoC3 excess also can cause high triglyceride and normal cholesterol level. This ApoC3 is associated with the increased interference of lipoprotein lipase enzyme, and therefore there will be deficiency of lipoprotein lipase in body. This ApoC3 excess is often associated with diabetes mellitus patient, and for this fasting plasma glucose test should be done to rule out diabetes mellitus. And the next category is diseases which associated with high HDL level. And in this case, we can found the cholesterol ester transporting gene defect, which is associated with cholesterol ester transfer protein deficiency. This leads to HDL-C level is more than 100 milligram per DL, and it is also associated with raised ApoA1 level. Next slide, please. The diseases associated with high cholesterol with high LDL, non-familiar polygenic hypercholesterolemia. This disease is very common, and it is seen that approximately 85% population suffering from hypercholesterolemia is have this non-familiar polygenic hypercholesterolemia disorder. And the laboratory finding shows that they are associated with high LDL cholesterol in serum. Another variety, and as this is a non-familiar variety, it is seen that less than 10% of first degree relatives are suffering from hypercholesterolemia. Second variety is familiar hypercholesterolemia. It is an autosomal dominant disorder caused by mutation of LDL receptor on chromosome 19. And this may be homozygous variety or heterozygous variety. In case of homozygous variety, the symptom develop in early childhood, and in heterozygous variety, the symptom develop for decade of life. For diagnosis purpose, in case of homozygous variety, LDL-C level is more than 400 milligram per DL is often found, and in case of heterozygous variety, LDL-C level is more than 220 milligram per DL is often found. And as this is a familiar type, first degree relatives are often associated with hypercholesterolemia. Now, if we consider more confirmatory tests, then we can do the LDL receptor measurement study. Or we can do the quantification of LDL receptor on lymphocyte surface by surface cell sorting technique like FACS. Now the more confirmatory test is DNA sequencing study. In DNA sequencing study, it is found that 4 common missense mutation is present in PCSK9 gene. Now the next type is familiar defective ApoB disorder. This mutation in ApoB gene in chromosome 2 is associated with this disorder. This leads to increased LDL-C level. Next slide, please. High cholesterol with high triglyceride level. The disease commonly found is familial combined hyperlipidemia. Though the genetic basis of this disease is unknown, it is thought that multiple genes is involved in this disease. Diagnostic parameters are high cholesterol level in serum with disproportionately high level of ApoB4. In serum, it is found the elevated small LDL particle is present, which justifies the presence of disproportionately high level of ApoB4. This beta lipoproteinemia. This disorder is due to homozygous ApoE2 genotype. ApoE gene may be a type ApoE3, ApoE2, and ApoE4 type. Among these, ApoE2 is least common, but if there is presence of homozygous variety of ApoE2 protein, then there will be defective LDL and VLDL uptake. This leads to abnormal increase in LDL and VLDL level in blood. For diagnostic parameter, we can find that VLDL and triglyceride ratio is raised, and it is often found that more than 0.3. For the normal VLDL and triglyceride ratio is 0.2, and a broad abnormal band is between VLDL and LDL found in electrophoresis study. This band is called beta VLDL. Genetic study also shows the E2E2 genotype for ApoE4. Now the fifth group, that is diseases associated with isolated low cholesterol level. There is two diseases. One is hypo-beta lipoproteinemia. Another is A-beta lipoproteinemia. Though hypo-beta lipoproteinemia is more common than A-beta lipoproteinemia. Hypo-beta lipoproteinemia is familiar in nature, and it is due to nonsense or missense mutation, and it is associated with increased cardiovascular risk. But A-beta lipoproteinemia is non-familiar nature. Therefore, if we do the LDL-C estimation of the parents of this disease, we found there is normal LDL-C level in case of A-beta lipoproteinemia, whereas in case of hypo-beta lipoproteinemia, this LDL-C level is very low among the parents. The homozygous variety of hypo-beta lipoproteinemia is associated with less than LDL level is less than 50 milligram per day. Now the last diseases are diseases associated with isolated low HDL level. This may be the Tangier disease and LCAT deficit. In Tangier disease, there will be a mutation of ATP binding cascade transporter A1. In homozygous variety of Tangier disease, we found HDL level is less than 55 milligram per kg. In case of heterozygous variety, we found HDL level is less than 30 milligram per dl in men and less than 40 milligram per dl in case of female. Genotyping study shows the mutation of ABCG. At present, more than 200 mutations of ABCG CA1G has been found. Now the last disease is lecithin cholesterol acyl transferase deficiency. This leads to partial or complete loss of LCAT activity, which leads to a lower HDL level less than 10 milligram per kg. and low plasma LCAT activity. So I conclude that the diagnostic parameter may be very common test, or may be associated with very sophisticated test. In case we need the very sophisticated test, we should refer the patient for with higher facility for this sophisticated test. Thank you.
Thank you, Dr. Haldar. In a nutshell, so you want to say that we should first rule out the secondary causes, then move on to the primary causes. And it involves an array of tests, which involves all the lipoprotein level detection, as well as genetic tests also. So thank you so much. I would this is just a request because we are pressed for time. I would like to request all our panelists to keep their answers very crisp and short. So my next question.
Dr. Priyanka, please unmute yourself. You're muted.
Okay. So am I audible again? I'm so sorry. So Dr.
Yeah, you're audible.
Soumyamoy Das, apart from screening and diagnostic tests, what supportive or ancillary tests can the clinical laboratory offer so far as long-term management of IAMs are concerned? Dr. Soumyamoy Das.
Yeah, thank you, Dr. Priyanka. As we know, the inborn error of metabolism are genetic disorders. Due to the defective production of the enzymes, there is defect in the pathway of metabolism. Whether it is anabolism or catabolism, there is a defect in the pathway. So, along with the treatment after diagnosis of the disease, most of the cases need lifelong treatment. So, along with lifelong treatment, the disease, the affected baby or person need lifelong monitoring. So, in these cases, there are so many tests. I will mention few, like blood gas analysis. Arterial blood gas analysis can be done in routine laboratory. From blood gas analysis, lab physician can diagnose whether the patient is suffering from any metabolic acidosis. As most of the organic acidemias in patients show metabolic acidosis with high anion gap. So during the treatment that is prescribed this test. Other tests are blood glucose, or plasma ammonia, or plasma amino acids, serum electrolytes, liver function test. And there is a list. Everyone can go through this. So by this test, the physician can monitor the patient whether there is any difference in the parameters, whether the treatment is effective, and if there is any disbalance or is imbalance, then physician can prescribe to increase or decrease the doses of the treatment. Next slide, please. So in the next here, I will discuss few of the tests like complete blood count with differential count. In organic aciduria, there is anemia, thrombocytopenia, or leukopenia or leukocytosis. So when the patient is taking treatment for this organic aciduria, the CBC and DLC should be done regularly. And if the patient is suffering from vitamin B12 or cobalamin or folate absorptive or intercellular utilization disorder, then these tests, this parameter should be see in the CBC. Like if the picture comes in megaloblastic anemia, then clinician can assume maybe further treatment or increase in the dose of the treatment is needed. There is renal function test. Normally, if the serum creatinine is high, physician can diagnose that the kidney is affected. But if the creatinine value is very low, there is this may be due to the decreased production or defect in the transport of the creatinine. So from renal function test, also this test can also be used as a monitoring test of the patient. Other is the liver function test. In liver function test, there are so many parameters. Like in Dubin-Johnson syndrome or Crigler-Najjar syndrome, if the patient is suffering and there is the treatment is not effective, then there will be raised bilirubin. So from liver function test, the clinician can easily monitor this specific disease. Next slide, please. Other few parameters like uric acid, high level of uric acid seen in the Lyss syndrome, or even it is also high in the glycogenolysis or gluconeogenesis cases. A low level may point towards xanthine or hypoxanthine disorder or a molybdenum cofactor deficiency. So if the patient is suffering from the defect in the ammonia metabolism, and then this uric acid test is very helpful for as a monitor tool. Other parameters as creatine kinase. Elevation of creatine kinase is seen in the glycogenolysis or rhabdomyolysis disorder of energy production, like fatty acid oxidation defects or gluconeogenesis defects or mitochondrial disorders. Next slide, please. Here I will mention few specific tests for the specific disease, like the disease tyrosinemia. In tyrosinemia, there is increased production of tyrosine. The disease is diagnosed by the presence of succinyl acetone in the urine sample, and the patient is prescribed a diet containing low tyrosine. And during the treatment period, regular monitoring of the tyrosine amino acid is required, and also there is requirement of the testing of the succinyl acetone in the urine. If the succinyl acetone is dissipate from the urine, then a physician can say that treatment is effective or treatment patient needs continuation of the treatment. Though in tyrosinemia, the treatment required for the lifetime, and measurement of alpha fetoprotein is also required in this disease, as most of the cases may develop hepatocarcinoma in the advanced lifecycle life. Another condition is phenylketonuria. In phenylketonuria, the patient is given phenylalanine-free diet, and in this patient, tyrosine is also given as tyrosine is synthesized from phenylalanine. So, along with the phenylalanine-free diet, the monitoring of the phenylalanine in the serum is required for better understanding the condition of the patient. Next slide, please. Another inborn disease is maple syrup urine disease. In this disease, patient required the regular plasma monitoring of the level of branched-chain amino acids, like the leucine, isoleucine, and valine. These are branched amino acids. In this disease, there is defective enzyme, so there is accumulation of the branched-chain amino acids. So by monitoring these amino acids, physician can say the treatment is better going on, or prognosis is better. So we have seen that certain substances in different body fluids can be used as indicator of both the presence and the progression of the disease, as well as for monitoring of the efficacy of the therapy. So other parameters, as I mentioned, two other parameters in this aspect, like creatine kinase or CPK. It is a non-specific indicator of metabolic myopathy, as it is found in mitochondrial fatty acid oxidation defect, in glycogen storage disease, Pompe disease. And nowadays, to cytokines, these are fibroblast growth factor 21 and growth differentiation factor 15. These are recently added as a potential biomarkers of mitochondrial myopathy. If the patient is suffering from this disease, then monitoring of this biomarker is effective. So we have seen that for the long-term monitoring of inborn error of metabolism, routine biochemical tests are like liver function test, kidney function test, or all the routine biochemical tests are required for to assess the overall biochemical changes in the patient's body in response to the therapy. So as the disease is for long time, so long time monitoring of the patient is needed by this routine biochemical test or special disease specific test like the detection of the tyrosine or phenylalanine or other markers in the bloodstream or urine. Thank you.
Thank you, Dr. Soumyamoy Das, for that insight. Now, let us move on to our next question. The next question, please. The next question is to Dr. Manidip Chakraborty. So, Dr. Manidip Chakraborty, welcome. What should be the standard protocol while addressing the nutritional requirements of a hemodynamically unstable septic patient of inborn errors of metabolism?
Thank you, Dr. Priyanka. Actually, most of the cases, the babies with inborn error of metabolism present in the emergency department in a very devastating manner. So initially, the treatment has to be started empirically without any diagnosis based on the clinical suspicions and the clinical pictures. So nutritional intervention in the acute phase is very much crucial and very much goal-oriented, what I mean to say. So first, the first goal is that we have to stop all those nutrients which may precipitate or which trigger the disease. That is the first and very much important thing. So we have to eliminate all those proteins, harmful proteins and sugars, that is galactose and fructose, that may trigger up the disease. And actually, the metabolic screen broadly classify or broadly categorize the patient of a IEM. So depending on the positive screen, we also take it out or we also off the disease-specific offending agent. So our second goal is that to stop the ongoing catabolic process. This is very important. If we allow the catabolic process to goes on, so what will happen? The noxious metabolic pathway will set in, and so toxic accumulates, the toxins or the toxic metabolites will accumulate in the body, and subsequently that will cause the deterioration of the patient or deterioration of the baby. So to promote the anabolism and to stop the catabolic process, we have to provide a high calorie. This calorie has to be provided through dextrose or in some cases through a lipid solution. Actually 10% dextrose that can be used as a dose of 10 to 12 milligram per kg and as an infusion. And sometimes if we use the intralipids or lipid emulsion solution for IV solution, then 0.5 gram per kg per day, that solution also can be used. So our third goal is that to combat with the hypoglycemia. This is very important. If the baby comes with a hypoglycemic emergency, so promptly the hypoglycemia that to be corrected, because otherwise there will be a devastating, there will be a catastrophe will occur. So actually the hypoglycemia that to be combat with the 25%, 10% dextrose, or in some cases if the patient is having central line along with the 25% dextrose as a dose of 0.25 to 1 gram per kg per dose, that to be used as a bolus to combat the hypoglycemia. So along with that, the other factors, other cofactors are vitamin that also to be infused like carnitine, vitamin B12, vitamin B6, biotin, that also to be infused to the patient. And after the screening report has come, so depending on the disease, we have to infuse some amino acids or some vitamins that to be administered. Like in urea cycle defects, we have to administer the arginine. We have to, as the arginine hydrochloride as the infusion, but in sometimes as a carbamyl phosphate synthetase deficiency or ornithine carbamylase deficiency, we have to reduce the doses. There are some other infusions that to be used like that, say glycine or carnitine is to be used as a specific manner. In secondary phase, after the acute management is over, the acute nutritional intervention is over. In secondary phase, we have to assess for the nutritional supplement because acute management is over. Now, secondarily, we have to think for the nutritional supplement. So, all the baby to be assessed for the very first day for the nutritional supplement. So, the supplement can be done but the assessment can be done based on the Nutrix score. This is a very unique score to be used. It denotes the metabolic need, the nutritional need of the patient. So if the patient is tolerates enteral food, if the patient tolerates enteral food, next slide please. If the patient tolerates enteral fluid and enteral feed, and though the patient is hemodynamically unstable, so enteral food to be continued. That is very much important thing. But the monitoring for the enteral food, that to be done, say after starting the enteral food, whether there is a clinical deterioration or not, whether the blood lactate level has increased or not, whether there is a abdominal distension or not, because blood lactate level directly proportional to the lactic acidosis or gut ischemia. So that to be monitored first. There are so many commercially available enteral food preparation are available in India, but these are very much expensive. So what can be done? That can be prepared based on the caloric exchange that freshly can be prepared. So if the patient tolerates enteral food, we have to give enteral food. If not tolerating, then we have to think for the total parenteral nutrition. That is very important, because after all, after the acute management, we have to add the nutritional supplement, because in IEM, this nutritional supplements or nutritional intervention is a very important issue. It's the mainstay of treatment. So in case of TPN, in the screen it is there, the caloric need that to be determined by the indirect calorimetry, and based on the type of that is very important. What is seen in the screen, that is a general formula for total parenteral nutrition, those who doesn't tolerating the enteral food. So if depending on the type of IEM, the food had to this formula has to be modified. That is very important. The formula has to be modified. So to conclude with that, so I must say that this nutritional intervention is very critical and very crucial in the case of management, in the management of the hemodynamically unstable patient with with sepsis and IEM. So we have to be very careful and we have to be very cautious whenever there is a question of nutritional intervention in case of IEM. Thank you.
Thank you so much. So a goal-oriented approach, stopping noxious nutrients and giving a proper nutritional supplement. So that forms the crux of treatment of such sick patients. Yes. Okay, thank you so much. We move on to our next speaker. Please, the next question is to Dr. Koushik Kumar Das. So what are the challenges or difficulties that might be faced by clinical laboratories while screening for or confirming the diagnosis of common inborn errors of metabolism? Dr. Koushik?
Yes, thank you, Dr. Priyanka. Am I audible?
Yes,
am
you
I
are
audible? Hello?
Yes, you are audible. You are audible. Please carry on.
Okay. 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 So the question here is directed towards the difficulties and challenges of newborn screening in clinical lab. Next slide, please. Next slide, please. So we can broadly classify all the difficulties and challenges in two categories. One category involving the difficulties which are related to interpretation of results, and the other category is all the challenges associated with the newborn screening. Now, the difficulties in interpretation of test, they can be the test or the screening test, they can yield false positive results as well as false negative results. We'll look on to this just in a moment. And the associated challenge, they are related to the handling of samples, the integration and collaboration which is much required, and obviously the economic concerns. Next slide, please. So first of all, the difficulties in interpretation of screening test. Now, as I have said that the screening test, they can yield false positive as well as false negative results. Now, there is a high rate of false positivity in the screening test. Now, as a result of this, we require additional confirmatory testing, targeting a more specific analyte using a different or more specific method. So there is a need for second tier testing. Now, in case of, as I have already mentioned, apart from false positive, there are also false negative results in cases of screening tests. Now, there are two things which I would like to mention, that the concentration of analyte, they can vary according or vary with respect to the newborn age. Say, for example, in cases of homocystinuria, the concentration of methionine, it can vary with the newborn age. So proper sampling or proper sample collection should be taken. And pseudo deficiency is another term related to false negative results. Now, what is this? Now, in cases of inborn error metabolism screening, we often detect the enzymatic activity by subjecting the normal enzymes to synthetic substrates. Now, what happens in certain cases? Say, for example, a person is normal, that is, he or she is having the normal enzyme for a particular metabolic pathway. When it is subjected to the normal substrate or natural substrate, there is a reaction. But when it is subject to synthetic substrate, it does not provide or does not produce any reaction. So that produces pseudo deficiency, and that yields the false negative results. Apart from these two, certain X-linked diseases like X-linked adrenoleukodystrophy, as this disease is X-linked, so the females are generally carriers of the disease and not affected. 50% of the female carriers of X-linked adrenoleukodystrophy, they actually do not show elevated levels of the marker, that is lysophosphatidyl-choline. So that is also another thing to mention in cases of difficulties in interpretation of screening test, as those 20% of the female carriers cannot be detected with the help of screening test. Next slide, please. Next slide, please. Yes. So next comes the associated challenges in newborn screening. It starts from the handling of samples, beginning from the collection as well as the transport to the lab. Well, all of this requires trained personnel and logistics. So that is obviously a challenge in cases of newborn screening. Those samples during collection, they should be collected at appropriate newborn age. Otherwise, they will yield a faulty report. And as a result of this, there are sometimes they yield frequent unsatisfactory results. So this has to be taken care of. Secondly, which I would like to mention is that newborn screening is not like, say, we undergo a screening test and we detect something and we say, yes, he's having this inborn error of metabolism. It is not like that. During newborn screening, there should be a collaboration and integration between public health, screening and diagnostic labs, physicians, and the families of the affected children. Now, without this integration and collaboration, the newborn screening program will not be efficacious. And last but not the least, the challenge is obviously an economic concern, because as my co-panelists have already discussed, that the screening tests are not sufficient. It requires confirmatory testing, second-tier testing, and further molecular and genetic testing as well. So that also raises the economic concerns. And also, these inborn errors of metabolism, they need follow-up. So these raise the economic concerns. So these are, in a nutshell, all the challenges or difficulties which we encounter during newborn screening. Thank you.
Thank you for that crisp but comprehensive presentation. My next question is to Dr. Bidyut Bhuyan. Dr. Bhuyan, can you provide us some insight on the more prevalent futuristic diagnostic methods for IEMs?
Yes.
Dr. Bhuyan.
Thank you for the question, Dr. Priyanka. Can you please move on to the slides here? So presently, the diagnostic modalities are done with the help of mass spectrometers. So mass spectrometry is an analytical test by which we can isolate a compound on the basis of mass and charge. So mass spectrometry have different types, and and the best modality here which helps us is the tandem mass spectrometry. So can you please move on to the next slide? Next slide, please. Okay. So doing this mass spectrometry, the sample which we collect for this instrument or the machine must be collected during an acute metabolic decompensated state. If it is collected in some example like fatty acid oxidation disorders, if it is collected after the episode, then it becomes undiagnosable. And suppose if the condition is, the lab is far away or the condition is not feasible presently, it may be stored. The urine or plasma or serum sample may be stored at -20 degrees centigrade for the later testing. And if everything is not there, suppose in a PHC or in a remote village, then at least a blood spot on a filter paper may provide enough material for investigation in emergency situation. Please, next slide, please. Next slide. Yes. So here we can see in the first picture, there is a mass spectrometer machine, tandem mass spectrometry, which gives the result in the computer screen. So let us briefly see how the mass spectrometry works. So any sample we inject into the machine, first it has to be charged. I know that charge is through the form of ions, in the form of ions. So there is an basically the electron, one of the chamber, the electron disintegrates molecules into smaller pieces, and then this ion source may be something called the MELD. Mass MELD is there. It is the matrix assisted laser desorption ion, or it may nowadays newer is used is the ESI, that is the electron spray. In this the ion sample. And there are a series of chambers, electric chamber, and ultimately the mass gets separated on this molecular and in the.
Dr. Bidyut, your voice is breaking up.
What is tandem mass spectrometry? Tandem mass.
Dr. Bidyut, your audio is breaking up. Dr. Bidyut, are you there?
Hello.
Dr. Bidyut?
Am I audible, ma'am?
You're audible, but
Yes,
your audio was breaking up.
Am I audible?
Yeah, you're audible. You continue now. Continue.
Maybe the internet connection was
Yes.
broke
That's
up.
fine. Continue.
Sorry,
Continue.
Okay. Yeah. So now let us see, understanding the mass spectrometry. Now let us understand a basis of tandem mass spectrometry. Tandem mass spectrometry is actually two mass spectrometers which are placed back to back within nearby position. So what we do is that ion source is coming, then it passes through the first mass analyzer, then it is like keeping a gatekeeper in the gate, or gatekeeper in the gate, or maybe in a club we see big people are there, bouncer type. So it removes other ions which are not necessary and keeps only the ion of interest. Suppose we are interested in glycine, example. So it will keep, we do such a, in the software, that only that particular ion is left. And there is again the activation chamber is there. In the activation chamber, it further breaks down the glycine
Dr. Bidyut? Dr. Bidyut? Dr. Bhuya, are you there? Dr. Bhuya?
Hello. Hello, ma'am. Hello. Am I audible?
Yes, you're audible. Please make it
Okay.
crisp and short. We are really pressed for time. I'm sorry to
Yes,
interrupt
yes.
like this. Yeah, please carry on.
So again, it breaks up all the breaks up into millions of pieces, and it measures all the pieces. And with the help of a 3D software, it creates its own mass. And then in this way, we can get a perfect picture of that particular compound or the molecule. So next slide, please. So here we can see the basis of tandem mass spectrometry. See, one, it's like a small puzzle. It is broken down into many pieces, and then each piece is measured within the detector, and then we can go ahead and create it 3D with the help of 3D software. We can create the mass, and we can pinpoint the actual compound. What is the compound? So next slide, please. So the main apart from this, we have already discussed. I have already discussed about the DNA testing, DNA sequencing, all the method genetic tests. So but the major limitation of DNA sequencing is that the suppose one enzyme is there, any enzyme, multiple genes are involved in creating one enzyme. So we may miss out some particular gene, and the study is incomplete. So what to do? So for example, methyl malonic acid, acidemia, this compound is created with the number of many genes. Many genes are involved in it. Or maple syrup urine disease, many genes are involved in for this creation. So next slide, please. So the gold standard in the future is coming up is the gene panel study. We take the important genes which are concerned for a particular enzyme, and then we take all the, suppose the methyl malonic acid, 50 genes are involved as per our knowledge, or 20 example, just an example. So we'll study each of the gene, and we'll create a picture, and we'll get the exact defect of that, pinpoint the disease or the gene, and hence we can comprehensively, with the help of biochemical and molecular investigation, hand in hand to confirm the diagnosis of a metabolic disorder. So in a nutshell, we can say that gene panel study is the future of IEMs. Thank you, ma'am.
Thank you, Dr. Bidyut. Thank you very much. Due to technical glitches, your talk got interrupted in between. So yes, sorry for that.
Sorry,
yeah, no, it's not your fault. It's just a technical fault. So we move on to the penultimate question of the session. And this is for Dr. Somnath Haldar. So do you feel the lab physician can play an active role in the arena of genetic counseling, and how? Dr. Haldar.
Yes, thank you, Dr. Priyanka. I personally think that when a laboratory physician can perform a very important role in case of genetic counseling for explaining how he or she can perform this important role. Please go to the next slide. Next slide, please. As day by day, it is found that non-communicable diseases are commonly associated with familial in nature. From this concept, the genetic counseling has become the core element of medical care system. Genetic counseling is a process of helping people to understand and to adapt to the physical medical and psychological and familiar implication of genetic contributor of the disease. Laboratory physician should work as a core member of genetic counseling process, and he or she should provide the adequate risk assessment, and also should provide the education support and other support to the individual and the family who have been diagnosed with the genetic disease or at the risk of development of genetic disorder in future. Laboratory physician responsibility is not only interpretation and disclosing the genetic testing results, but also providing the adequate counseling support. They should act as an advocate of patients with genetic disorder. Next slide, please. So, in nutshell, the laboratory physician can involve in genetic counseling process by following ways. He or she can explain the inherent pattern of a genetic disorder to the patient and patient family. He or she can also explain the risks of recurrence of a genetic disorder in future. And laboratory physician can also perform the carrier testing, especially for couples based with associated couple of consanguineous family. Laboratory physician can also perform carrier screening tests for the extensive carrier identification. And the laboratory physician also should also perform the psychological counseling to the patient and patient family, as many of the genetic disorders may not be so much so much problematic to the patient, such as phenylketonuria. In case of phenylketonuria, if we give the phenylalanine restricted diet, the symptom of phenylketonuria may not develop in future. And if the patient party knows about this information, the depression of the patient party will be relieved. Now, once the diagnosis is confirmed, the report should be dispatched to the concerned physician with proper guidelines and for appropriate carrier identification process and possibility of a recurrence of the genetic disease in the family. Now, overall role of laboratory physician in genetic counseling. The laboratory physician may have diagnostic role and also have carrier testing. The laboratory physician can perform biochemical and genetic testing for detection of the genetic disorder, and also can detect the risk of recurrence of a disease in future. The laboratory physician can also perform genetic counseling of the patient and patient relatives regarding the future, regarding the genetic disorder prospect in future. The laboratory physician can also perform carrier testing of the patient and the sibling of the patient and the partner of the patient and the parent of the patient. By doing this, laboratory physician can easily identify the risks of development the genetic disorder in future sibling of the family. Thank you.
Thank you, Dr. Haldar. Thank you for providing us with that insight. And now for the last question, it is never easy to be the last speaker. So Dr. Manidip Chakraborty, my question to you is, can you enlighten us regarding the current policy that has been adopted by the government of India for screening of inborn errors of metabolism? Dr. Manidip Chakraborty, over to you.
Thank you, Dr. Priyanka. At the very beginning, I would like to highlight that the government of India yet to have a mandatory screening program for the newborns to detect the inborn error of metabolism. If I go a little flashback, so newborn screening is still a young science that has been coined by Professor Robert Guthrie in 1960. And after that, in 1980, it has been started first in Bangalore. And after a long time lapse, the Goa government has been started in 2008, and subsequently the government of Kerala and the government of the Union Territory of Chandigarh has been started, and all these three states are running these programs of screening of the newborn screening with a grand success in respect of the lowering infant mortality rate. They even lowered the infant mortality rate even lower than the country as a whole. But earlier, in the meantime, the government of India also has launched the project name that is RBSK, that is Rashtriya Bal Swasthya Karyakram, with the help of National Health Mission. Basically, it is a child screening and early intervention program that is widely acts on 0 to 18 years of age, acts on the children age group of 0 to 18 years. Usually, this group of children are usually screened for any birth defect, deficiencies, or any disabilities, and they are accordingly the action is taken for by this project. But the thing is that the detection newborn screening for IEM has been overlooked, or this is disfocused actually. Focus has not been given in this project. Whereas in the past decades, since the past decades, the awareness regarding NBS has been increased like anything. Very private sectors also, they are doing newborn screening, and even the Delhi government under RBSK, of course, they have been started a project like it is called as NEEV, that is Newborn Early Evaluation Vision. Under NEEV, they are planning to cover 1.5 lakhs of children or 5 lakhs of newborn to detect, to tackle with regarding the say functional, visible, and the metabolic defects. And they have been started already, and in between ICMR also they have been given emphasis and given they said that it is very much necessary that the newborn screening to be done for few of the diseases. And as per MARD India organization data, currently Indian government also, it is a very good news that Indian governments also, they have formalized the rare disease policy, and newborn screening for IM is given a immense importance on that. As a part of this, I am proud to say that Indian government have started a project as an initiative very recently that is named as Ummid. It's really a Ummid, or really a hope to all those patients or all those families, to them say treatment or diagnosis of IEM is like a daydream. In the Ummid, they will take all the project will be funded by the central government of India, and in this project, what they will do, they will tackle all those genetic say inherited disorders, and including IEM. So they are having a aim actually Ummid, that is the unique method of management and treatment of inherited disorders. Actually, they have founded, they have started this with a aim to establish Nidan Kendra. They have been started, there are certain aims and objectives. The Nidan Kendra, that is National Inherited Disease Administration. They are planning to start with a state-of-art genetic laboratory with expert doctors in genetic in gene therapy and expert technicians. There will be these kendras will help in screening, screening and prenatal screening, also in diagnosing and management of those cases. In a single language, I can say they will exert a multidisciplinary care to all those patients. There's another aim is that to produce the skilled technicians and skilled doctors. So they are planning to produce skilled doctors, and in a say per year they are planning to produce on 96 doctors, and this time span they have been decided initially the time span should be for 3 years. So per year they will be producing 96 doctors and the required amount of technicians, so that the genetic laboratory or genetic diagnostic laboratory that can be set in the even district level also. So the another thing is that another aim of Ummid is that to undertake the pregnant screening, to undertake the newborn screening, including the IEM in the district level. They will adopt a district hospital. They are planning to start, along with a full-fledged genetic laboratory. They are planning to give. They are also planning to spread awareness among the clinicians, so that the more number of cases that to be screened. So it's a really ummid to the lots of people, of the poor people of the India. They to them there is treatment and diagnosis of IM is like a daydream. And lastly, I will conclude that basically for a newborn, the best gift by a nation is a healthy future, and proudly say that India is going to give a healthy future. And who knows, I pray and hope that the government of India will very shortly start the mandatory newborn screening, and of course they will give a healthy future, of course a healthy civilization. Thank you.
We all pray along with you, Dr. Manidip, that government of India realizes, and we do hope that they soon start this screening program. So that was the last question of the session. So from this session, we came to know what are the broad classifications of inborn errors of metabolism, that there should be proper policies for diagnosing and for clinching the diagnosis of inborn errors of metabolism. Also, MS, NMR, metabolomics, which is a very upcoming thing, they form the mainstay of the diagnosis. Gene panel study is a futuristic approach. And for therapy, it should be more goal-oriented, and noxious stimuli or noxious metabolites should be taken out from the diet. And the child should also receive proper nutritional supplement. Enteral feeding should be instituted for early on, and genetic counseling should be given to the prospective parents. And then policies that have been adopted by government of India, which are very upcoming things, Umeed, Nidan, and RBSK. And I am ending this session for the day with a note that yes, we pray and hope that government of India and we as a nation give our children, who are the future of the nation, a bright and healthy future. That's all. Thank you so much.
For the question regarding the nutritional supplement, that has to be given for lifelong, because long-term nutritional management is the mainstay of treatment of those IEMs. So of course, diet, the specified diet that should be given to the patient, and of course for that awareness is very much required to the family of those patients. And depending on the prescribed diet, that should be started lifelong. That should be continued for lifelong.
Thank you. Next question is for Indranil Chakraborty, Dr. Indranil Chakrabarti. The question is, which is the most common IM disorder in Indian scenario? The question is open to others also. The question is open to others.
Hello.
Hello, ma'am. Can I answer the question?
Yeah, sure. You can answer.
Yeah.
Sure, you can answer.
Talking from the Indian, yeah. So, am I audible, ma'am?
Yeah, audible.
Am I audible, ma'am?
You can speak.
Yeah.
Yes.
So,
you're audible.
yeah. So talking, yeah. So talking from the Indian perspective, I think as malaria is very much endemic in different regions of India, so and different studies show that there is strong association between the glucose 6-phosphate deficiency and the endemicity of malaria. So I think G6PD or glucose 6-phosphate deficiency is the most common inborn error of metabolism in Indian scenario. Apart from that, certain organic aciduria like methylmalonic aciduria, propionic acidemias, they are common in India. Thank you.
Thank you. Thank you. Next question is for Dr. Bidyut Bhuyan. Okay. What should be the ideal time for sample collection for the newborn screening test?
Thank you, ma'am. Actually, ma'am, it depends. In the Indian scenario, I cannot say because there is no compulsory program for newborn screening in India. But as per USA, it depends from 24 to 48 hours to 72 hours. And they actually, if symptomatic baby is born, then they challenge the baby with protein diet or some challenge test is done. And as soon as they cleanse the diagnosis, then screening is started, ma'am. It is screening is done. But as per Indian scenario, ma'am, I cannot say because no compulsory program is present in our country. But outside countries, there 24 to 72 hours. It depends, ma'am. 1 to 3 days.
Okay.
They do the screening.
Okay. Thank you. Thank you, Dr. Bhuyan.
Thank you.
Next question is for Dr. Somnath Haldar. Your question is, since we are talking about the genetic counseling, who will guide and motivate the family for genetic counseling?
Okay, ma'am, as far as genetic counseling is concerned, the laboratory physician who initially diagnose the genetic disorder will make the initial counseling to motivate the family of the affected child, and he or she will also guide the family for performing a carrier testing of the family, and also guide for risk review of the genetic disease. After that, he can refer the patient and the family for more expertise genetic counseling to a psychiatric counselor or any other person.
Okay, thank you. Thank you, Dr. Haldar. So last question of the session is for Dr. Manidip Chakraborty. Since we have talked about the UMID program and the NIDAN,
Yes.
is it possible for the UMID project to UMID initiative of the government to go establish in each and every government setup or each and every government medical college?
No, ma'am. That will take time because there are so many barriers out there. That will take time. Initially, right now, 5 Nidan Kendras are running throughout India. So, ma'am, the main district level, they will adopt each and 7 Nidan Kendras that will adopt one district hospital. The plan is like this. So, ma'am, each and every district hospital to cover that will take time and may or may not be possible, ma'am.
Okay. Okay. Thank you, Dr. Manidip. So this
Welcome.
brings to the end of the question answering session. Thank you all the panelists for your presentation and your answering processes. And thank you, Dr. Priyanka, the moderator, who really did her job very well. And
Thank you, ma'am.
thank you all our ACCLMP members and the organizing committee who have come so far to bring this program a success. Thank you all.
Thank you, ma'am.
So as we have
Thank you.
come to the end of this program, I would like to thank all the attendees who have made this program a great success. Our panelists, our young brigade, including the moderator, who has really actively participated and enlightened us with their excellent discussions on inborn errors of metabolism, which everybody has gained something or the other from their discussions. I also thank our Student Affairs Chairperson, Dr. Madhurima Bora and Dr. Shayantan Dasgupta for their inputs. To name the mentors who has done so much for making this program a success, I have to name, of course, Dr. Sharmishta Chaudhary, who has tirelessly done so much to make it a success. Backstage, she has till late night, she has worked hard to bring our youngsters to the forefront. I also thank all the mentors, namely Dr. Anindya Dasgupta, Dr. Shushut Sen, Dr. Madhumita Das, Dr. Santoshmita, Dr. Barnali, Dr. Mohsina Ma'am, Dr. Teresa, Dr. Mrinal, and last but not the least, and we also have backstage workers like mentors like Dr. Shireen Gogoi. Last but not the least, I thank the person behind every success of these webinars we have been conducting is our web designer, Mr. Anil, and we are looking forward for more such webinars in the near future. Thank you. Thank you all.
Thank you, ma'am. Thank you so much.
Thank you, ma'am.
Thank you, ma'am. Thank you so much.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Great presentation, team. Thank you so much. Thank you all. Thank you, Dr. Koushik, Dr. Soumyamoy, Dr. Bidyut, Dr. Indranil Chakrabarti, Dr. Manidip. Great presentation.
Thank you, ma'am.
Thank you, Dr. Priyanka Moitra.
Last but not the least, Dr. Somnath Haldar. I really missed his name. I'm so sorry.